Welcome to the second module on plant stress responses. Today we will be covering the molecular aspect of the response. So in today's lecture we will go over gene expression, stress hormones and growth regulators, basic temperature responses for heat and cold, plant defense response, and pathogen resistance. Let's have a closer look at this figure. We can see that a stress response is initiated when plants recognize stress at the cellular level. Stress recognition activates signal transduction pathways that transmit information within the individual cell and throughout the plant. Changes in gene expression may modify growth and development and even influence reproductive capabilities. Plants respond to stresses as individual cells and as whole organisms. Stress-induced signals can be transmitted throughout the plant, making other parts more ready for withstanding the stress. Alteration in gene expression can result in increase of amount of specific mRNA, enhanced translation, stabilized proteins, altered protein activity, and a combination of the above. This scheme shows overlapping and complex cellular responses resulting from abiotic stress. These responses are a recurring theme throughout the chapter, as responses to many stresses overlap, sharing what we can call transcriptional hubs. Primary stresses such as cold, drought, heat, and cold result in secondary stresses such as altered osmotic or ionic potential. The cell senses initial stress signals such as osmotic and ionic effects, temperature, membrane fluidity changes, and then triggers downstream signaling cascades and amplifications of the signal. The final layer is the transcription factors, which initiate stress-responsive mechanisms in order to protect the cell and restore homeostasis. In the following section, we will go over plant stress regulators, more specifically hormones. Those include abscisic acid, jasmonic acid, salicylic acid, and ethylene. Plant hormones are signal molecules produced within the plant and occur in extremely low concentrations. Hormones regulate cellular processes in targeted cells locally and moves to other locations in other functional parts of the plant. Abscisic acid, also called ABA, is one of the most important plant growth regulators. Abscisic acid owes its name to its role in abscission of the plant leaves. ABA is produced in the roots in response to decreased soil water potential and other situations in which plant may be under stress. ABA then translocates to the leaves where it rapidly alters the osmotic potential of stomatal guard cells, causing them to shrink and the stomata to close. This reduces transpiration and water loss. Under cold stress, ABA slows down cell division in the vascular cambium, adjusting plant cells to cold condition and protecting dormant buds from the cold stress. Plants that are treated with ABA will be somewhat cold hardened. Here is a schematic representation of transcriptional regulatory network of cis-acting elements and transcription factors involved in abiotic stress responses. Transcription factors are shown in ellipses. In this example, an abiotic stress activated signal transduction pathways that use 4 different types of regulons, which are transcription factor networks, to activate acclimation mechanisms. The regulons shown belong to the MYC, MYB, BZIP, DREB, and NAC families. For each regulon, the name of the DNA cis element bound by the transcription factor is shown. Cis acting elements are shown in boxes, and target stress inducible genes are shown in a long rectangular box at the bottom. Jasmonic signaling can be induced by a range of abiotic stresses, including osmotic stress, wounding, drought, and exposure to elicitors, which include chitins, oligosaccharides, oligogalacturanoids, and extracts from yeast. Jasmonic acid biosynthesis in Arabidopsis is also regulated by cues in the developing stamen, where jasmonic acid is required for pollen development. However, we do not yet know how these stresses or developmental cues are perceived. One approach has been to search for the earliest response to stress, which would therefore be a candidate for a component of stress reception signal transduction pathway. In this figure, we can see the local and systemic responses to wounding mediated by cysteamine and jasmonic acid. Procysteamine is proteolytically processed to the peptide hormone cysteamine, which activates synthesis of jasmonic acid from linoleic acid. Jasmonic acid stimulates the transcription of genes encoding proteinase inhibitor PIN2 and other wound response proteins and in remote tissues to which jasmonic acid is transported. Salicylic acid is a phytohormone that plays a major role in photosynthesis, transpiration, ion uptake, and transport, which controls plant growth and development. Salicylic acid in particular influences seed germination, seedling establishment, cell growth, respiration, stomata closure, senescence-associated gene expression, and responses to abiotic stresses. Its effect on some of these processes may be indirect because SA alters the synthesis of and signaling by other plant hormones including jasmonic acid, ethylene, and auxin. In addition, SA functions as a key signal in regulating thermogenesis and disease resistance. SA is involved in signaling and mediating in plant defense against pathogens. It plays a role in the resistance to pathogens by inducing the production of pathogenesis-related proteins. It is involved in the systemic acquired resistance, SAR, in which pathogenic attack on one part of the plant induces resistance in the other parts. Ethylene is produced from essentially all parts of higher plants, including leaves, stems, roots, flowers, fruit, tubers, and seeds. Ethylene production is regulated by a variety of developmental and environmental factors. During the life of the plant, ethylene production is induced during certain stages of growth such as germination, ripening of fruits, abscission of leaves, and senescence of flowers. Ethylene production can also be induced by a variety of external aspects such as mechanical wounding, environmental stresses, and certain chemicals including auxin and other regulators. Some of the environmental stresses that can induce the production of ethylene include drought, flooding, mechanical pressure, injury, and infection. Ethylene is required to mediate both resistance against necrotrophic fungal pathogens and against soil-borne fungal species that are not ordinarily plant pathogens. This figure is just an example of how these signaling hormones work together in response to a pathogen or a wound. You do not need to know these pathways in detail. It is only important to know that these hormones do work together in response to pathogen defense in order to cut down on energy expenditure from the plant. In the next two slides, we will talk briefly about cold and heat stresses. We will go into more details about these stresses in our future modules. As you remember, Protein structure is sensitive to disruption by changes in temperature, pH, or ionic strength associated with different types of abiotic stresses. A heat stress response is induced by temperatures between 10 to 15 Celsius above normal. When a plant is in a state of heat stress, a unique set of chaperones called heat shock proteins are synthesized in response to a variety of environmental stresses. Chaperones are proteins that physically interact with other proteins to facilitate protein folding, reduce misfolding, stabilize tertiary structure, and prevent aggregation or disaggregation. Heat shock protein induction is mediated by a B-zip factor. In the case of a cold temperature stress, cells increase the accumulation of osmolytes to retain water and stabilize proteins in the cells. Some examples on those stabilizing proteins include proline and glycine betaine. Under cold temperature stresses, cells undergo changes in gene expression. Those can alter the production of many proteins such as antifreeze proteins, proteases, RNA binding proteins. Many cold-regulated promoters have DREC elements. Like animals, plants have a number of ways of protecting themselves against diseases. Their first line of defense is their outer layer of tissue, the epidermis or cork, which is generally covered with a barrier of waxes, cutin, or suberin. However, if a pathogen penetrates this barrier, the plant responds by producing other protective molecules. Plants and pathogens have evolved together such that pathogens have mechanisms to attack and penetrate plants, and plants have evolved mechanisms to kill the pathogens and limit the infection. The following slides will show different plant defenses a plant goes through to protect itself from pathogens. A few of the major pathogens are discussed in this slide. A plant can be attacked by not only insects but also many different pathogens such as viruses, viroids, bacteria, fungi, and nematodes. It is important to know the differences between these pathogens in order to correctly identify the cause of the problem. Always know your crop and how it can be infected in different ways. Pathogens can be virulent, which means causing the disease in a plant because the plant has little or no genetic defense, or it could be avirulent, which means that it could cause mild harm but not death. In the figures, we can see a tobacco mosaic virus, which is an example of a single-stranded RNA virus, and we can see a cauliflower mosaic virus, which is a double-stranded DNA virus. And now let's have a look at 3 different pathogen attack strategies. In the first one, necrotrophic pathogens kill the tissue of the plant and then colonize it. Necrotrophic pathogens have a broad host range. An example on necrotrophic pathogens are rotting bacteria. The second type of strategies belongs to the biotrophic pathogens. Biotrophic pathogens keep their cells alive, but they have a narrow host range, most likely one plant species. Examples on that would be viruses, nematodes, and fungal mildews. The third pathogen strategy is hemobiotrophy, where pathogens initially keep cells alive but kills them in later infection. In response to biotic stresses, plants develop some defense barriers that can help them protect themselves from those pathogens. The first barrier is the physical barrier. It is considered the first line of defense of all plants, where epidermal cells throughout the plant secrete wax to protect the surface from water loss and attack. Examples of a physical barrier include cuticle, thorns, and cell walls. The second barrier is the chemical barrier, where many plants produce toxins that kill herbivores, make them ill, or repel them with strong flavors or odors. Injured plants send out chemical signals that may signal other plants to prepare for an attack or attract other insects to eat the insect that are attacking the plant. And lastly, they can produce those chemicals to act as poisons, repellents, or to affect the herbivore's digestive system. The third barrier is the induced response, also known as plant defense response. The plant defense response or the induced response have three aspects: hypersensitive, local, and systemic. In the hypersensitive response, the first line of activated defense occurs within 24 hours, where a genetically incompatible pathogen is recognized and the plant then creates an unfavorable condition for the pathogen growth and reproduction. Following this strategy, the plant impairs the spread of harmful enzymes and toxins, and then follows a localized cell and tissue death. In the local response, alterations in secondary metabolic pathways can result in cessation of cell cycle, synthesis of pathogenesis-related proteins, production of ethylene and jasmonic acid, or fortification of cell walls. In the systemic response, defense responses are elaborated in tissues from the invasion site and even in neighboring plants. Plant disease resistance protects plants from pathogens in two ways: by performed mechanisms and by infection-induced responses of the immune system. Relative to a susceptible plant, disease resistance is the reduction of pathogen growth on or in the plant, while the term disease tolerance describes plants that exhibit little disease damage despite substantial pathogen levels. Disease outcome is determined by the three-way interaction of pathogen, the plant, and the environmental condition. Defense-activating compounds can move cell to cell and systemically through the plant vascular system. However, plants do not have circulating immune cells, so most cell types exhibit a broad suite of antimicrobial defenses. Now let's have a look at this video that explains the Floer's gene-to-gene resistant hypothesis.
Big A, big R, resistance, disease. Oh man, how can I figure out the G4G model? Oh man, what should I do? Oh no way.
Oh, hey Tom, how's it going? Are you ready for the test?
No, I cannot make it. I cannot figure out G4G model. Look
Really?
at this.
Oh, why? That's really easy, man.
What?
Yeah, I figured it all out by watching this
Really?
video on YouTube.
Uh-huh.
Yeah, it's this old black and white movie. I think
Really?
it was made around the time that H.H. Floor came up with this hypothesis.
Okay,
Do you want to watch it with me?
sounds good.
Yeah, let's
do
watch it. So, what did you think of it?
Yeah, that really makes sense. I just have one question. Why did the owner keep a dog so the dog can recognize any intruder?
Oh, that's a really good question.
Mhm.
And it's funny you mention that because plant pathologists have already discovered that that kind of system
Really?
already exists in plants.
Okay.
Yeah, they call it the guard hypothesis.
Guard hypothesis. Wow.
Yeah, but that's not part of this test.
Is that weird that the people in the video is look like us?
The recognition of the pathogen by the R gene product leads to hypersensitive response. The hypersensitive response is a mechanism used by plants to prevent the spread of infection by microbial pathogens. The HR is characterized by the rapid death cell of local regions surrounding an infection. The HR serves to restrict the growth and spread of pathogens to other parts of the plant. The HR is analogous to the innate immune system found in animals, and commonly preceded by a slower systematic or a whole plant response, which ultimately leads to a systematic acquired resistance. Fungi, bacteria, and viruses activate systemically a specific subset of PR-type gene by a mechanism known as systemic acquired resistance (SAR), in which local necrosis formation at the initial site of pathogen invasion triggers both a local increase in SA accumulation and the formation of fluid-mobile signal. A hypersensitive response will also set up signal transduction pathways that results in systemic acquired resistance. Recall that the activation of the SAR response requires accumulation of the endogenous salicylic acid, which is triggered by the presence of a pathogen. In Arabidopsis, a high concentration of salicylic acid activates a molecular signal transduction pathway that is identified by a gene called NEM1, also known as NPR1 or SAI1. The pathway results in a heightened immunity to all pathogens in uninfected parts of the plant, sometimes for many days after the attack. Because the SAR elicits production of various phytoalexins and PR proteins, it tends to be nonspecific and can afford protection against a variety of pathogens. Here's an overview of plant defense against an avirulent pathogen. In number 1, we can see a plant protein, a receptor, and a pathogen protein, a ligand, join. This causes a signal transduction pathway to be initiated. The HR response occurs in number 3, where cells seal off pathogens and then undergo apoptosis. Dying cells release a chemical signal, salicylate, and salicylate is transported to other parts of the plant where signal transduction pathways are initiated in distant parts of the plant. SAR is achieved when a phytoalexin and PR proteins are produced on those remote tissues. This following video shows us responses that a plant performs in order to limit an invading pathogen from infecting the entire plant.
Plants and pathogens have co-evolved such that pathogens can recognize plants by the sugars and other molecules they produce. Plants and pathogens have co-evolved such that pathogens can recognize plants by the sugars and other molecules they produce. Plants, in turn, can recognize pathogens by the molecules they produce. The ability to recognize pathogens allows plants to activate defense systems that can prevent widespread infection. A plant may recognize molecules of a pathogen directly or indirectly. Some pathogen molecules, for example, bind directly to plant receptors in the plasma membrane. Others, such as enzymes, break down molecules within the plant's cell wall, and then these broken down products bind to plant receptors. When molecules bind to the plant receptors, these receptors become activated and trigger a cascade of reactions. These reactions lead to the production of second messengers, molecules that migrate through the cell and induce it to respond. Here, the cell responds by expressing certain genes. After certain genes are expressed, the plant begins to produce a class of molecules called phytoalexins. Phytoalexins are usually phenolics or terpenes, and a cell produces them within hours of the onset of infection. These molecules can kill a variety of invading pathogen species. Some of the genes that are expressed code for proteins called pathogenesis-related or PR proteins. Some PR proteins kill bacteria or fungi by breaking down their cell walls. Others may travel through intercellular passageways called plasmodesmata and serve as alarm signals to neighboring uninfected cells. When a plant encounters the pathogen, one of its first responses is to produce polysaccharides. These polysaccharides can strengthen cell walls, making it harder for the next pathogen to break through. They can also plug plasmodesmata, preventing certain pathogens, such as viruses, from migrating to neighboring cells. A plant may also use a mechanism called the hypersensitive response to prevent widespread infection by an invading pathogen. In the hypersensitive response, infected cells essentially commit suicide, producing a patch of dead tissue called a necrotic lesion that walls off the pathogen. A phenomenon called systemic acquired resistance may then follow the hypersensitive response. In systemic acquired resistance, the entire plant increases its resistance to a wide range of pathogens. In one model for how this occurs, a compound similar to aspirin called salicylic acid is produced during the hypersensitive response. In the model, salicylic acid is transported from the infected tissue to the rest of the plant body. Salicylic acid induces cells throughout the plant to produce PR proteins, which then lie in wait to kill any pathogens that might enter the plant. These PR proteins immunize the plant against a variety of pathogens.
And finally, we reach the conclusion. In response to abiotic stresses such as drought, salinity, heat, cold, and mechanical wounding, many genes are regulated and their gene products function in providing stress tolerance to the plant. Understanding the molecular mechanisms of plant responses to abiotic and biotic stresses is very important as it facilitates in exploiting them to improve stress tolerance and productivity. By the end of this lecture, you should know the answers to the following questions. What are the gene expression outcomes under stress conditions? Name the plant hormones involved in stress regulation and what are their roles? What are the different pathogen attack strategies? What are the plant defense strategies against pathogens? How are pathogens recognized by the R gene product? What is plant systemic acquired resistance?